first, I want to express my appreciation to Stem Cell Congress and the Stem Cell Congress Committee and the effort for this Congress. Today I want to talk about stem cell and isolation of stem cell from dental pulp. Let's have a background about this field. Successful isolation of mesenchymal stem cell from dental pulp might be extremely promising for developing stem cell-based therapies. Dental pulp stem cells can be used in regenerative endodontic therapy. The aim of this study was to introduce a method for dental pulp stem cell isolation. Mesenchymal stem cell isolated from bone marrow are the most intensively studied adult stem cells. Since the procedure for harvesting bone marrow stem cell is painful, invasive, and cause complication in up to 30% of patients. So, the search for alternative stem cell source has been started. Stem cells are considered the most valuable cells for regenerative medicine. Stem cells can be classified as either embryonic or postnatal stem cells. Both can contribute to the development of regenerative medicine. There have been many advances in postnatal stem cell research. Newer studies suggest that adult stem cells may have much greater plasticity than previously thought. There are various sources of stem cells in adult tissue, such as bone marrow, blood, brain, the eye, skeletal muscle, liver, dental pulp, skin, the lining of the gastrointestinal tract, and pancreas. Adult stem cells are rare, difficult to identify, purify, and culture. They can be classified as a hematopoietic or mesenchymal stem cells. Bone marrow-derived mesenchymal stem cells have been the most studied mesenchymal stem cells. However, it's unclear if these cells have the capacity for dentinogenic differentiation. Bone marrow-derived mesenchymal stem cells have been the most studied mesenchymal stem cells. However, it's unclear if these cells have the capacity for dentinogenic differentiation. Actually, Grontos and his team were the first to isolate dental pulp stem cell in 2000. Further investigation revealed that these cells have intensive capacity of differentiation into neuron-like cells, adipocyte, osteocyte, and chondrocytic cells. Dental pulp stem cells demonstrated a higher developmental potential and longer lifespan than bone marrow stromal stem cells. This has been found that the number of proliferating cells as well as the frequency of colony-forming cells were significantly higher in dental pulp stem cells when compared to bone marrow stromal stem cells. On the other hand, dental pulp stem cells can be cryopreserved and used for regenerative purpose later. Based upon this finding, dental pulp stem cells may be considered appropriate cell type for developing tissue engineering strategies. The stem cell population in the pulp is very small. Approximately about 1% of the total number of cells are stem cells, and the effect of aging reduces the cell pool available. Exfoliated human deciduous tooth contain multi-potential stem cells that identified to be a population of highly proliferative and colonogenic. These cells are capable of differentiating into a variety of cell types, including neural cells, adipocytes, and odontoblasts. The goal of the present study was to design a dental pulp stem cell isolation method with high in vitro expansion potential and optimize the culture conditions for their increased proliferation. And let's talk about method and material of this study. In our study, we used 40 primary teeth that was exfoliating used in this study were removed from 30 healthy patients aged between 6 to 9 years old. Informed consent was obtained from the patient's parents after receiving approval by the Institutional Ethics Committee of Isfahan University of Medical Science. The teeth were immersed in sterile phosphate buffer saline (PBS) stored on ice pack and immediately transported to cell culture lab for sample processing. After cleaning the surface and disinfect with iodine, a horizontal groove was cut along the cemento enamel junction, used diamond fissure wire with high-speed handpiece and copious water supply on a high-speed handpiece to split the teeth and obtain the pulp tissue under sterile condition. Here we have a figure that shows digestion of pulp segments with collagenase dispause enzyme that destroyed pulp scaffold. All pulp tissue were minced into small fragments with a fine scalpel. The fragments were digested in a solution of 1 milligram per milliliter collagenase dispause for 30 minutes at 37 centigrade and centrifuge at 500 for 5 minutes. Cell suspension were seeded in 60 millimeter culture dishes contain minimum essential medium alpha modification called alpha MEM with 20% fetal bovine serum and 100 penicillin and 100 microgram streptomycin and also 1 microgram per milliliter amphotericin B. Here we after 1 day we cultured the stem cell, and at day 1 we have stem cells here. After 2 days the stem cells were garrowed, and after 3 days. The cells were counted and their viability was determined by trypsin blue staining. The results demonstrated a few cell colonies were found within 4 days after initial plating. Cells were homogeneous fibroblast-like in morphology. We sought to examine the expression of nucleosome OCT4, JMJ2C, and nano-G as proliferating and self-renewal regulatory factors for stem cells. Based on the RT-PCR and agarose gel electrophoresis, the cells from all groups express all the genes. Here we have the cells after passage 1, and we see these stem cells after passage 2, and finally after passage 3. So, expression of stem cell marker, we used 4 marker in PCR, as I mentioned, nucleosome, JMJD2C, nano-G, and OCT4 in the stem cells. And finally, conclusion. In conclusion, the result of the current study indicates that adult dental pulp contain stem cells that can be isolated and expand efficiency. These cells are probably one of the source of regeneration after pulp injury. They could be valuable in cell-based therapeutic approach too. This study show that pulp contains cells with high plasticity and proliferation capacity and can be easily isolated without any serious intervention. And finally, I want to invite all of you to our beautiful and historical city. It's Isfahan in Iran, and there are many old and historical bridges and also palaces in our city. So I invite you to visit our city. Thank you for your attention.
Any comments or questions? I have some. I think this was fantastic, and I'm very surprised that you're so versatile, because it's better than bone marrow.
Yeah.
Would you be proposing to use it for other things than teeth?
Actually, if we used primary teeth, the stem cells in primary teeth are more potential than adult teeth because they are near to the in the young age, and the stem cells are more potent than adult teeth in secondary teeth. It depends on the age of the patient. So we recommend to use stem cells in primary teeth.
Okay, so you're using this is primarily for dental purposes.
Yeah.
You could use it, from what you said, for other, instead of bone marrow.
Mm-hmm.
Use it on bone as well?
Actually, we want a method that can isolate stem cell without any harmful procedure. If, for example, a 9 years old boy, that his teeth is exfoliating, it's not, we don't do anything harmful for the patient, and we can get the stem cell from this good source.
And would you use it for, instead of filling children's teeth, would you use stem cells to fill?
Maybe in the future we can use stem cells in dentistry for producing a new teeth. But not yet.
Not yet. And how long do you think that will be before that happens?
I think 40 to 50 years.
Not bad though, is it? But all dentists are at work.
Yeah, of course.
Okay, thank you very much.
You're-
What's comment here?
Were there any differences at an age-specific basis? You studied samples from 6-year-olds to 9-year-olds.
Uh-huh.
Was there a difference across that age?
Actually, we don't compare the age of the patients. We use this age because at this age, patients' teeth, primary teeth, are exfoliating. So we get the teeth and sectioned and get the pulp. Thank you.